We divided the class in 3 groups. The water group, working with invertebrates. The invertebrate group, working with dragonflies. And the plant group. Even if you don't see it, there are millions of microorganisms living in the water. Our mission today was to see the invisible.
Here we are in the heart of Serbia, on the hike to nowhere, but we're going somewhere anyway, and we're gonna have a great day.
During our field trip, the first step was going to the pond. In the pond we learned how to sample, and we raised 3 different samples where we put 3 different tickets, writing down the name, the date and the location. Afterwards, we also went to the river, raising other samples of microorganisms living underwater. Here, the water has a very high speed, and these organisms are very different and are more resistant to water speed.
The surface, they take the light, and there is not too much light underneath. So maybe, for instance, on the beginning of the season, this wasn't so much covered, and the same type of plankton lived. For that, we will use this. This is basically a net, because it's made from silk, and it has a mesh size like somewhere around 20-25 micrometers. So it's made to basically you put water inside, and it releases water, and in this little cup it keeps all the organisms that are here.
The samples will then be placed in a falcon tube.
Why would we sample plankton organisms from some water body?
To see the density of the population of the ecosystem.
And
so we took some water from the lake, 10 liters, to put it inside the plankton net, so it can filter the water, so we can collect the organisms that are living in the lake. We took 3 samples of the organisms. We put them in falcon tubes with 5 to 10 milliliters of alcohol, and then we put the dates and the place of collect, so we can analyze it tonight.
The techniques you just witnessed are part of a field called limnology, the study of stagnant waters or waters with very slow currents.
Now you've just seen what we did today, but there's plenty of things that we didn't explain, and we are going to do it now.
Just as a reminder, we wanted to know how to qualify the water and how to detect pollution inside the water.
In the second sample, we used a kind of research that is called plantanology. It's the science of study the river with floating water. We studied Baltic organisms that live in the rock at the bottom of the river because the water is more static. How to use these organisms as indicator of pollution quality?
We have an indicator, the Baltic organisms, but we know how to use this indicator. For that, we have a technique called trend biotic index. First, you took the sample in the previous video, and now we have to see under a microscope the different groups of the Baltic organism. So now we have to see the difference and who are the different groups of the organism.
Lots of group of Baltic organisms, but we're focused on one characteristic, the sensibility to pollution. So some organisms are pollution sensible, like for example the Plecoptera, the Ephemerptera, the Tridoptera, but others can be resilient to pollution, like Coleoptera, Iridinea, and Oligochaeta.
Now we can complete the table in function of the resistance of each group. Which cell I am talking about, you'll see now.
As we said before, there's 3 taxonomic groups which are more sensible to pollution: Plecoptera, Ephemerptera, and Trichoptera. This is related to their score. So the more sensible to pollution, they will have the higher score. The less sensible to pollution, they will have the lower score. So in our sample, we will see which taxonomic groups are present, and we will add their score. The higher the score is, the lower the water is polluted.
Now we know how to function the table. So we can have an example with our own sample we make all the day. So let's go.
Of our second sample result. So what we can consider is we get a caddisfly. A caddisfly is a Trichoptera, an insect, and it's composed in 3 segments. The first segment is the head. It's outside the house it constructs. So we can see the house here. It's composed of sand and rocks. And the second segment and the third segment, the abdomen and the hook, are protected by the house. The hooks serve to be attached in order to not be thrown away by the current. Also, here we can see a main line.
Our results have shown that in the water there was the presence of Plecoptera, Ephemerptera, Trichoptera, and Diptera. And these, as stated before, are all symbols of great water quality and very pure water.
Yeah!
As a limit of the day, we have to say that we didn't work in a good sterilized environment, and that we didn't analyze the first sample because we didn't have the time to put the sample with the bacteria and the plankton in the spectrophotometer to analyze it.
Aquaviva, Szobrak, dobry wynik.